announcements. So I think, as most of you on the call are aware, the AMP ASCO CAP Guidelines Committee has recently released an updated draft summary of the changes to the recommendations for the upcoming V2 guidelines, and it's currently open for community review. Now, one of the things we want to do as a community that's, of course, closely associated with those guidelines, we want to make sure that we are hearing the feedback of our members and ensuring that we're providing that feedback in summary form from our organization to AMP. So if you have feedback on the AMP ASCO CAP guidelines, we recently sent around a survey, and by recently I mean this morning, maybe about an hour or two ago, um, asking for feedback on those guidelines, which we're going to collate and then submit as an organizational response. So, if you'd like to provide your feedback as part of the VIC organizational response, we'd be happy to have it. It's a way to maybe multiply some of your individual perspectives in addition to the individual feedback you may provide to the AMP guidelines. So I'd encourage people to do that if you have an interest, and we'll be taking feedback for the next week and then submitting those comments by the indicated deadline of February 28th. So we'd appreciate everyone's participation if you have interest or comments on those recent draft changes to the guidelines. Okay, so with that said, it's my distinct pleasure to welcome Elaine Mardis. Elaine's been with us, of course, on this call before and is a well-known figure in our community. But we're delighted to have her here today to tell us about the progress and plans of the Molecular Characterization Initiative and the work that we're doing to advance that. So, Elaine, I will hand it over to you, and we'll take questions after your talk.
Great. Thanks for the intro, Alex, and thanks for accommodating me when I had to cancel in January due to sort of recovery from illness, which made me think I wasn't going to be able to make it through the talk without coughing out of control. So feeling much better now, and I'm happy to be here today for the purposes of this presentation. So I'm going to share screen here and go to the right title slide, and hopefully this is visible and you can hear me. All
Looks
right,
good.
thumbs up. Thank you very much. So as Alex mentioned, I'm going to be talking about the MCI as we refer to it. Feels like I've been doing projects sponsored by the NIH for a long time, and we always have to have an acronym to go with every project. So this is the one that we've been using for a while now to describe this very focused effort that I'll tell you about today. I do want to give a little bit of background, as is typical, maybe telling you all things that you already know, but not everyone is so up to speed on pediatric cancer. So, I just wanted to provide a little bit of background and also to give some background on the work that we did here at Nationwide Children's Hospital in a local cohort that really ended up being a practice run for the MCI, and I think poised us well to respond to this very large scale project. So pediatric cancer, here's a fact. Cancer is the leading cause of death by disease in the developed world in children over 1 year of age. Most children with a cancer diagnosis are diagnosed with acute lymphoblastic leukemia, but in fact, the leading cause of death by cancer is brain cancer. And I'll focus a little bit on that today, but not exclusively. I think the other thing to point out, which people may or may not be aware of, is really the challenge of pediatric cancer, where if you take all of pediatric cancer in aggregate, the cure rate, if you will, is commonly cited at about 80% across both solid and liquid tissue malignancies. However, as pointed out in this sort of epidemiology-oriented paper from a few years ago in JAMA Oncology, the long-term side effects of chemotherapy, radiotherapy, and other approaches, including surgery, this is just focusing on chemo and radiotherapy, really has attendant long-term impacts on the health and well-being of individuals that survive their cancer. So this was just looking back through the decades, if you were diagnosed with cancer as a child in the 70s, 80s, or 90s, what were the sort of changes, if any, in a long-term cumulative mortality risk based on things other than cancer, but also including cancer, because we know that in the setting of care with radiotherapy or chemotherapy, survivors have an attendant significant increase in the likelihood of developing secondary cancers. So the bottom line is that we have improvement in cure rates and long-term survival of these individuals, even if you look, at this decade by decade approach. But the bottom line is there's still a significant risk of shortened lifespan due to a variety of factors. They're shown here, lung, pulmonary, cardiac, and also now a suicidality due to extensive and enduring pain. And also secondary cancers, which I mentioned. So I think we still have significant challenges in front of us. But one of the biggest challenges, which I'll focus on today, is the challenge of an accurate diagnosis, which has already been shown in large-scale studies to have an impact on some of these aspects here. So the other sort of, I think, known to many, but maybe not all, fact is that pediatric cancers are quite different than adult cancers. Far fewer point mutations. This makes sense because most pediatric cancers have not had a lifespan over which to develop and accumulate mutations like adult cancers. But also some challenges that really require more comprehensive sequencing of the DNA than in the adult setting, where you're typically seeing panel tests being used to identify hotspot mutations that then would correspond to some sort of therapy or would calculate a high TMB that would get you immunotherapy. So in particular, on the left-hand side of this slide, you see sort of the small overlap between significantly mutated genes done a few years ago by the German group coming out of their INFORM study, providing whole genome and transcriptome sequencing to kids across the country of Germany, and really showing that the commonly mutated genes, the gray area down below, there are some drivers that we know, but many of these are actually tumor suppressors. The profiling of normal DNA, of course, is also really needed in the pediatric setting, not for some of the reasons that we cite so often in adult cancers, but because genetic susceptibility, again, because these patients are developing cancer so early in life, often some children being born with cancer, we know that genetic susceptibility has a much more significant role. And as I will point out in the MCI, one of the things that we're also learning and was recently acknowledged in a paper by Kim Nickel at St. Jude is that often the adult susceptibility drivers like BRCA2, BRCA1, et cetera, that we often think about almost exclusively in the adult setting are being identified in kids. So I think we need to think about this more, and also maybe use different methods to get at more of the genetic susceptibility that we're probably missing. Another aspect that's really important based on WHO guidelines for the standard of diagnosis in brain tumors, but I think is seeking additional application across pediatric cancers, is the use of methylation profiling of the tumor DNA. And I'll talk about this extensively in terms of a classifier to use to train these data based on machine learning to provide an accurate and precise diagnosis. And then finally, one of the things that I've long been enthusiastic about is profiling of tumor DNA. This is important in the first case because many pediatric cancers are driven by gene fusions, as illustrated on the right, and I'll show you more about this in the context of the MCI, but also because I think we can learn a lot more out of RNA than simply identifying gene fusions and internal tandem duplications. So we can talk about that more if desired at the end. I did want to focus a little bit, though, on the therapeutic angles before we dive into the details of molecular profiling. And in particular, to just note that in that small area of overlap between drivers in the adult setting and in the pediatric setting, We do find now FDA approved drugs, for example, that target specific known fusion drivers in that overlap. NTRK fusions are probably very important in this regard because they have been so highly successful, especially brain penetrant and NTRK inhibitors in kids with essentially no hope of cure, providing very, very long-term and durable responses. And so these are just headlines from multiple registration trials for FDA approval of these drugs. We also have a significant opportunity, often unexplored in rare cancers, with small molecule targets. There's a beautiful study from the New England, Patricia Brastianis lab and others, sorry, Priscilla Brastianis lab and others, really looking at a very rare cancer type, papillary craniopharyngiomas, that are driven by BRAF mutations. Here, a dual targeting with BRAF MAC inhibition showed extraordinary responses, minimal toxicity, all the things, right, that you would imagine. But in a setting where there is no defined standard of care other than radiotherapy, it looks like this would be a much more preferable, lower side effect profile and apparently durable response to these simple simple small molecule inhibitors. And then finally, excuse me, from the group in Toronto and also from a worldwide trial, again, in a very rare tumor setting, children with high TMB, largely pediatric CNS malignancies, showing extraordinary responses to PD-1 inhibition. And essentially allowing this to become standard of care in a setting where there, again, are, sort of no new therapies beyond radiotherapy with a high degree of efficacy in many cases, and a relatively small number of children that have not achieved durable responses with these therapies. So some very interesting, just in the last few years, developments that make me hopeful that if we continue to press on some of the unique drivers now in children, we may be able to identify therapeutics that correspond to our highly precise diagnosis. So switching gears now to that theme, I'll just mention briefly that we had here at the Institute for Genomic Medicine a 4-year cancer protocol, which used multiplex molecular profiling, as you can see in this slide. Many debates about whether to do whole genome sequencing or exome, but at the end of the day, we sort of compromised because we wanted the high depth of the exome coverage to give us information about clonality and low level potential for driver mutations. But also because we recognized that it was really important from a prognostic standpoint in certain pediatric tumors to appreciate copy number changes across the chromosome. So arm or whole chromosome level copy number changes needed this sort of enhanced exome where we effectively combine standard IDT exome reagent with a copy number directed reagent that provides roughly 50 kb resolution across every chromosome arm and allows us to call copy number variants with a reasonably high degree of resolution. We also performed RNA-seq from the tumor, multiple analytics on this, as I'll describe in a minute. And then over time, we included the EPIC 850K DNA methylation array, really at the urging of our neuropathologists who encouraged us to think that prior to the 2021 WHO guideline changes, this was going to become part of the standard of diagnosis. So in those days, we used the DKFZ, CNS, or sarcoma classifiers. As I'll describe in a minute, that's now changed. And then to keep track of everything so that we could publish on the results of this protocol, we developed a REDCap database, which captured extensive information, including longitudinal clinical data on the cohort that would be required to report out in the peer-reviewed literature about certain aspects of the protocol. This infographic just shows you the uptake in general on this cancer heme protocol over the 4-year time period in terms of different patients that were studied, et cetera, and the total number being close to 500. I will point out, though, that in the course of this 4-year protocol, we were also approached by other provider groups within the hospital about the applicability of tumor normal exome sequencing in particular, and to some extent RNA-seq for the patients that they were caring for. And in particular, I'll point out here that vascular malformation and overgrowth syndromes enrolled a significant number of patients, where what we're finding when we look at tissue from these patients that go through surgical reduction surgeries, you know, we use that tissue high depth profiling, and we're able to actually find often somatic mosaic mutations in known cancer genes. PI3 kinase and mTOR in particular, pathway genes, are subject to mutations, especially in these areas of overgrowth. And in children with overgrowth syndromes, which has a significant burden in terms of psychosocial and other aspects beyond the fact that part of their body continues to grow, there are several FDA approvals now for therapies that were originally developed for cancer, but turn out to be, in many cases, highly efficacious for reducing the rate of overgrowth in these children and allow them to have a little bit more normal life. In aggregate, these all transitioned, the tests that were offered through this protocol all transitioned to the clinical setting in 2022. And in particular, our overgrowth and vascular malformation clinic patients also can be enrolled on the tumor normal exome comparison for a clinical readout of the drivers of the overgrowth or vascular malformation. This is just a final infographic to show that we really were able to demonstrate on multiple levels that we had diagnostic, prognostic, or therapeutically relevant findings from this protocol. Over 370 pediatric cancer patients studied, over 90% receive at least one of these defined medically meaningful results. So identifying cancer predisposition, finding fusions or targetable gene expression, a medically meaningful copy number variant that could be for prognosis, as I mentioned, or a druggable target, and then also somatic variants that were identified that were likely to correspond to an available FDA-approved therapy. We're in the process now and have been writing up the results from this protocol. They're emerging slowly. So we published on the germline susceptibility in the CNS malignancies, which, if you noticed on the previous slide, was the largest malignancy that was enrolled on the protocol. That got published last year. We're just getting a paper in resubmission around the infant cancers that were studied in this, and most recently accepted a paper on some of the heme malignancy patients. So I won't talk about those today, but I just want to use that as a precursor to talk about how we managed to scale this up and continue to scale it up for this large-scale MCI program that's funded by the National Cancer Institute and performed in collaboration with the Children's Oncology Group, as I'll describe. So this sort of crazy diagram briefly shows the flow of samples coming into this program, wherein patients being enrolled onto COG's Project Every Child Registry, which has been modified for the MCI, submit a tumor and normal match sample to our biorepository center here at Nationwide Children's. DNA and RNA are extracted, subjected to quality control, and then couriered over to our clinical laboratory. All of the intake of information is electronic for these samples, so that's where our laboratory information management system, which is LabVantage, takes the place of the accessioning of these samples and the corresponding clinical data, including a preliminary pathology report and reason for study. With this information in hand, then the sample analytes, the DNA and RNA, enter the laboratory, clinical laboratory for processing through. And you can see on the second bullet point that the clinical testing being performed here is tumor normal DNA exome, as I just described, For fusions, we're using a fixed panel called the Archer FusionPlex, and then the methylation arrays. And our job, not always so straightforward, is to sign out clinical results for these three tests within a 14-day turnaround time of having received the analytes from BPC. So their timeline is 5 days. Our timeline is 14 days. And we're really trying to get all of this information sent back to the ordering clinician within a 21-day total turnaround time so that it can be utilized for patient care in real time. That could mean getting them an FDA-approved therapy. It could mean enrolling them onto a COG or other clinical trial with the right molecular data in hand and satisfying other inclusion criteria. The best part, besides helping kids in real time with cancer diagnosis, is that all of the data, as I'll talk about, then gets subsequently auto PHI redacted in our computer systems here within the LabVantage LIMS and submitted to the Childhood Cancer Database, which is an NCI hosted public data repository. Our stipulation in the contract is within 90 days of test results. In actual fact, this all happens in the cloud. So we joke that it's actually within 90 seconds of test results because literally the PHI redaction happens quickly after the signed out report is communicated. Here, we're also delivering the results using the cloud. So the provider gets an email that the test results are available. They sign on to the system with their CTEP secure sign-on, and they can immediately download the clinical report for further consideration, either in molecular tumor board or in making treatment decisions directly. Ultimately, all of these data will be combined with digital pathology images for each patient and an integrated sort of final diagnosis, if you will, that will also be available within the childhood cancer database. Of course, it's all PHI redacted, so the individual reports, the digital path, and the diagnosis information are all connected with the same unique sample identifier, which is not indicative of the patient's identity, but allows one to do additional data mining. And that's really, to me, one of the huge values of this program is that we're generating a very large amount of data, as you'll see, that can go forward for additional impact on pediatric cancer in the future. Here's just a slightly more crazy diagram that describes all of the things that I just mentioned in terms of the entry of biospecimens on the upper left-hand side. You can see that we have now expanded this roster for solid tumors eligible for MCI to CNS. That was where we started. So brain cancers, sarcomas, neuroblastoma, is NBL, and then rare cancers is a very large basket of rare cancers that are eligible for this study. And then everything follows from what I said, essentially. So we have the analysis, return of results within 21 days, et cetera, and ultimately at the bottom, integration of data within the CCD data repository for future studies in pediatric cancer. Computationally, all of the data that are generated for these different assays that we're performing go directly to our Amazon cloud computing environment. The pipelines are Dockers that are based there. And as you can see on the right-hand column here, the different types of results that come out of those pipelines for further consideration by our variant analysts, variant scientists, and our clinical directors prior to sign out. Because this is a program that's reaching out, as I'll show in just a moment, across the United States, including New York State, we have to have all assays validated by CAP/CLIA, as well as New York State Department of Health. And so this has been the ethos since we rolled out this program for enrollment at the end of March 2022, which was when we accepted the first patient in, and things have kind of gone crazy since then. But I want to talk about that right now. What I wanted to focus on just for a second is sort of where we've had to get to just based on some of the crazy stuff that goes on when you start something and want to run it for 5 years and hope everything stays the same. So in 23, Illumina notified us that the 850K arrays were no longer going to be available. They were going to be changing those to 935. And this was their so-called version 2, and that rendered our ability to use the DKFC classifier that we currently had in our Docker pipeline not usable for diagnosis. Upon further discussion with the DKFC, it was pretty clear that they were going in different directions, and so we wouldn't be able to count on using that classifier either. And so it fell to us to also come up with an appropriate classifier for the version 2 arrays, and we did that. That's a publication that we're working on right now, so hopefully we'll be seen in print relatively soon. But we developed what we call the MacDaddy classifier. That's not just a funny name, however, it is an acronym that stands for something, and that is shown here: Methylation Array Classification, Diagnostic and Data Integration. Thanks to one of our clinical directors, Jesse Hunter, for coming up with that. And this is for central nervous system tumors. So you see the impetus here. I've already described it, the change in arrays and the change in the DKFC model. And so whereas DKFC has used a random forest algorithm fueled with now tens of thousands of samples, our choice based on Qi Chen, who was the primary driver of the development of this classifier, he and Marco Leung, who was the clinical director that worked closely with Qi and others to bring this to reality, are cited at the bottom of the slide on the right-hand side. So Qi decided to use a regularized generalized linear model machine learning approach to train the classifier. I don't really understand what that means, but he could tell you. But basically what we needed to do was to be able to put together enough data from the world of EPIC version 1.0, the 850K array, of which there was a lot, and the version 2 arrays, of which there was not much, to really be compatible with both array formats, but importantly, to be able to accurately classify tumors coming through the version 2 arrays. And this was validated in comparison to version 1.0, and also using the DKFC online classifier for version 2.0 arrays. And we had a go live for this validated classifier in May of 2024 in concert with the switch to the version 2 arrays. Here's just a little bit of information about how he was able to do this. So using a lot of original data sets in the IDAT format, which is unique to the Illumina methylation arrays, created a training data set using RGLM approach and cross-validation, and then looking carefully at those results and their ability to predict CNS in different tiers of classification: superfamily, family class, and subclass. And then we had a clinical validation data set of samples that we had run in duplicate on version 1 arrays for the clinical test for MCI, and then the remainder on version 2 arrays, so that we could have that comparison to identify how well the classifier was doing. And this went through multiple iterations. It uses the same 4-level classifications as the DKFC classifier, so the clinical reports look part and parcel the same as they would if they were coming out of the prior pipeline. Some advantages to this approach: the model training is fast. This takes under 4 hours. The random forest takes many days in most cases, and the internal cross-validation error rate was 0. Kind of hard to beat that. And we had, some discordancies, as you can see on this small table. One of the things that we did as a procedural decision on some of these discordancies, where we weren't quite getting down to subclass level relevance, is we realized that as the DKFC approach had evolved, let's just say, some of the subclasses that were coming out were not really clinically relevant. And so what we made a decision to do was to have these come back to clinically relevant terms referring to the WHO guidelines as opposed to the DKFC classification. We felt that that would be more germane to physicians, oncologists, and pathologists who are looking at these results than these subclasses that are often not meaningful in terms of the patient's diagnosis or treatment. And so this is just a quick screen view of what the results look like coming out of the clinical form I just point out two things in addition to the classifier, which is just an example shown here that scores with a very high score as a diffuse glioma with IDH mutation and a 1p19q retained astroglial class. We can show on the left-hand lower part of the report MGMT methylation. This is a methylation array after all, and we can look specifically at those sites on the chip to report whether MGMT is methylated or not. This has treatment value in terms of the use of temozolomide or not. And that is a clinical result. Then in the box, two pieces of research information that actually turn out to be pretty helpful, but are not being reported clinically. One is a copy number variation profile across the genome. This is, again, tumor DNA, so it shows us alterations in copy number at reasonable resolution genome-wide. We can compare that further on with the somatic tumor normal exome with copy number probes that I mentioned. And then finally, a t-SNE plot that's active. It's interactive on the electronic version of the report, where you can see where the patient under study is grouping relative to a large database of other similar diagnoses across CNS malignancies. And that can also provide help when you're looking at samples that don't necessarily exactly fit into a comfortable classification. Just to give you a brief summary here, as of the end of January for CNS malignancies through the MCI, over 2,800 cases signed out. About 86% of those actually had results that provided a clear classification based on the MacDaddy classifier. So I just want to show now some brief vignettes on the overall MCI program that will hopefully be of interest and just give you an idea of the breadth and scope of this program as it's reached out across the United States and in some cases across the world. So here, these are just numbers about different assays and the number of samples that have been received in, again, starting at the end of March 2022. Now over 11,500 orders signed out within that 14-day turnaround time. A Herculean effort, I can assure you, and many of our clinical directors who are on this can also probably assure you more painfully than I can. But this is impactful, so it's really gratifying on multiple levels that it's so apparently worthwhile. These are just data from the end of last year, a little out of sync with the end of January, but you get the idea. So here, of over 4,300 patients enrolled and consented, this is now closer to 4,500. Of the 226 COG affiliated institutions across the globe, 206 have submitted at least one sample. Many orders received, signed out within turnaround time, and you can see that patients from 47 states have enrolled, some more than others. So Texas, California, thankfully Ohio is a big buy-in here. New York State, those are the ones in dark blue, in Florida. Some are gray. Kansas, I think, is taking a hit because Kansas City Children's Mercy has submitted lots of samples, but I think they all get credited to Missouri and so on. And I just went to Australia at the end of last year to talk about MCI, and indeed about 10% of the patients enrolled in MCI come from COG sites in Canada, Australia, and New Zealand. So this really has, I think, worldwide implications, offering profiling data to kids everywhere that in many cases would not have access to this information. This continues to expand, as you can see. So Ewing sarcoma will begin enrolling sometime in this quarter. And then I won't talk about this today, but we do have plans underway to ultimately replace the Archer fusion assay with RNA-seq assay. Those are in the works now. And really, I think, ultimately provide clinical results, again, here on fusion, internal tandem duplications, but ultimately a much richer data source going into the CCD, as I mentioned earlier, than the Archer results. Here's just a nice table looking at the differences across different solid tissue malignancies that are enrolling here. Again, this is as of the end of January, where we have CNS sort of leading the way in terms of total cases. But for the different headings on the table, what you can see are the relative percentages of germline SNV, germline CNV, and then total cases with the germline finding for each type of cancer collectively, and then on the far right, somatic results for those in terms of SNVs and CNVs. So I think you can take a lot of things away from this table. One of the things I think that's really interesting from the standpoint of germline susceptibility is that we are seeing, in some cases, relatively high proportional germline susceptibility in the rare cancers in particular. But I think there's probably some that's not found by short read sequencing. So maybe more work needs to be done on that in the future. In aggregate, close to 14% of patients with a reportable germline finding. I just, for the purposes of illustration, chose CNS malignancies. The numbers are highest here, but I'll just pace through these results relatively quickly to just show you in aggregate in this case, germline SNVs that are identified and the genes that are associated with those. So CHEK2 is really the predominant leader here, TP53. And then we're seeing a lot of low-grade gliomas, so you won't be surprised by NF1. Maybe one of the more surprising things is this ELP1 variants that were described a few years ago in a very strict setting, a subtype of medulloblastoma as a germline susceptibility. This was work from Paul Northcott's lab, but now look to be actually perhaps a bit more prevalent. And just recently, we saw a complete deletion of this gene. So what we had previously sort of seen were point mutations that were deleterious, but more recently a copy number missing, an allele missing for one of the patients that was under study. Similarly, for copy number variants, we see a lot of different cancer predisposition syndromes. You can sort of read through them here. A couple of examples of mismatch repair defects, constitutional mismatch repair, and Lynch, where these are patients that are going to be having a higher tumor mutational burden, and so unlikely to benefit from checkpoint blockade, as I was pointing out earlier. But certainly lots of reportable results for CNVs. And then here's the ticker tape for somatic SNVs, and you can also see CNVs about 76% return there on genes. These are genes. This is such a large number of patients that we're now to just reporting SNVs that were found in at least 26 patients. But here the clear leaders are TP53 and BRAF. But as well, H3/3As are histone mutations that are common drivers in high-grade gliomas. So a pretty good number of those just compared to the rarity of that tumor type writ large. So these are diffuse midline glioma, typically pilocytic, or typically in the pons, sorry. Finally, I mentioned fusions are being detected, and so this is just a summary across the different disease groups. Some of you who know neuroblastoma know that it's not a fusion-driven cancer, so we're not performing Archer on the neuroblastoma samples. We certainly will be on Ewing, but in this case, we're just seeing, again, differential proportionality for fusion drivers with soft tissue sarcomas sort of leading the way at almost 42% find rate. Unfortunately, many of these are not druggable targets, but we're certainly cataloging those. So I wanted to just sort of stop there and shift gears to the last bit of the talk and just show you a couple of examples of where we're going with all this. This is just a couple of slides courtesy of one of my colleagues here at Nationwide Children's, Dr. Genevieve Kendall, who's really focused on using the zebrafish as a novel model system for pediatric sarcomas. And the way that Genevieve's sarcoma modeling work has benefited from MCI is taking some of the novel gene fusions or gene fusion partners that are being identified from our work and introducing those into zebrafish. In some cases, actually creating models of specific fusion drivers where there has never been a model before, just for a variety of reasons. And so this is sort of a view that she put together for me for these purposes that articulates the different parts of the pipeline. You'll see the little green dots, and so you'll know on the lower left here that GFP expression is a part of the deal, so that these fish who are largely transparent can be evaluated in terms of their tumor growth under the appropriate fluorescence light. And here's just a couple of examples. So PAX7 FOXO1 is a pretty well-known fusion driver in this lower fish, but on the top, a novel fusion SRF NCOA2. And for your viewing pleasure, a little bit of the work that goes on in the isolation taking a look at these fish under the proper lighting, where you can see, hopefully, the little green dots swimming around that are representative of the fusion-driven tumors that are developing in these new model systems. In this case, a VGLL2 NCOA2 fusion, for which Genevieve informed me there has not been a model previously available. So kind of cool that we're able to more immediately translate this work directly into new model systems. And I'm excited that we'll actually be having a conference here in 2026 at our hospital, sponsored by the Company of Biologists, to focus on lots of novel pediatric models. So that will be coming up soon. I'm just going to finish up today by talking briefly about challenges to this 14-day turnaround. I'm not going to talk through all of these challenges. I've just sort of listed them out here so to illustrate kind of what they are and what we're planning to do about the different challenges on the right-hand side of the purple bars. But in particular, just in these last few minutes, I'll focus on the bottom two challenges. So just one slide briefly on changes to our library prep workflow. Really about 75 to 80% of the samples that come into MCI are from formalin-fixed paraffin-embedded tissues. That's not ideal for DNA or RNA, and so we have been working hard to identify and now validate a new library prep workflow that I'm just illustrating here in this one slide. Our current clinically validated workflow is from NEB, which is shown on the left. For FFP DNA, we have an input requirement of 500 nanograms, and that's because we really don't have the access to all of the DNA to make libraries out of using next generation approaches. It's really just the partially double-stranded and the fully double-stranded that are suitable for adapter ligation in this scenario. The alternative library prep that is now just almost at the point of validation is from a small company in Santa Cruz, Claret. And what the Claret approach does is illustrated here on the right-hand side of the slide, is effectively the first step in the process is to turn all the DNA extracted from the tumor mass into single-stranded DNA. So that allows you to encompass all of the different types of molecules that are present in the extract. Then subsequent adapter ligation occurs, and you have a library that gives you the complexity and ability to do high-depth sequencing, but starting from only 25 nanograms of input. And in fact, this is the input amount that we've used for our validation of this library prep. So we're pretty excited to get that introduced, and hopefully we'll see a significant uptick, not only in the number of samples that qualify into MCI, because there were some that were being left out just due to inadequate amounts of DNA, but also just much cleaner profiles available for variant calling. And then lastly, near and dear to Alex and Wes's heart, this, I think Alex actually gave me this slide, so you've probably seen it. And this is trying to address the real bottleneck that human evaluation creates. Not because of humans necessarily, but because we have so many data sources to go to really interpret and sign out these results successfully. And so we have a couple of things going on here that Alex and Wes have been working on. First, of course, and probably familiar to many of you, is the variant categorization tool, or VARCAT, which is really going to change our lives soon, I hear, which is sort of articulated by these bullet points on the right-hand side of the slide. Really meant to sort of not only aggregate and harmonize data, but also use a clinical variant classification framework that allows us to assign oncogenicity as well as the conventional ClinGen or AMP ASCO-CAP tiering to these, signed out results. So we're pretty excited about starting to use this. The programming, as I understand it, just wrapped up on Valentine's Day. Some user acceptance testing is going to be going on in the next couple of weeks and bug fixes, although I'm sure there are no bugs. But then we will do the introduction and validation with our clinical directors and variant scientists helping us with comparing this to our current approach. And then lastly, VARCAT and some other tools roll up into a larger system that we refer to as AVA, Automated Variant Analysis Toolkit, which really allows us to transition from the use of Excel spreadsheets. Thank goodness. I think this will address scalability in a big way over time, but really essentially builds a system where we can evaluate variants, include the ones that we want to include by using sort of a shopping cart system like you would with online shopping. So anything you want written into the report, you choose. At the end, you effectively check out, and this shopping cart feature will streamline the clinical report creation. Also, part of this, of course, is capturing the information from every patient into a large database called Barhouse and using that as part of the AVA system to inform us of when we've seen variants previously, how they were signed out, and also give us the ability to version the Barhouse so that as new information comes forward, we can understand for previously signed out patient samples what version of the system was used and what's new and different about the new version of that database. So this is sort of multiple parts that are connected together for variant gene and evidence curation using VICC and GA4GH standards-based data evidence normalization and harvesting. Then the local part of the system uses VARCAT, some new visualization tools, and the Barhouse database, which you can see down here at the bottom, including interaction with LabVantage for the purposes of the sign out for these patients. This is the system in which our sign out occurs. And then finally, and importantly, sharing of variant data with external sites and supports, global standards, et cetera. Ultimately, my hope for this system is that we're able to make it available to multiple users, not just within our own walls, but globally, so that we can really enhance the standardization, the capture of variants, and especially in the context of the germline variability to really begin to understand that better worldwide. But we'll see how that goes. That's sort of the long-term hope. Last comment that I just threw in at the last, and I think it's an interesting point for conversation and questions just as I wrap up, and that is the challenges now being thrown at us from different reports on saturation mutagenesis using CRISPR, primarily looking at VUSs that are missense variants in a variety of genes. So just within the last year, BAP1, just within the last few weeks, BRCA2, couple of papers in Nature, another paper looking at site saturation mutagenesis of 500 human protein domains, largely kinases, and then TP53 coming out of Thierry Soucy's lab and with multiple other people. So really now starting to ascribe functionality, if you will, using specific readouts following CRISPR mutagenesis that sort of saturates through the genes or regions of the genes, and using that readout system as a way to sort of start establishing benign versus pathogenic. I think it presents really interesting challenges in terms of clinical variant sign out for VUSs and how we handle that as a community moving forward. So I think interesting times. These are just some obvious conclusions about the need to combine DNA and RNA, the importance of profiling in pediatric cancers for maximum precision diagnosis and also for treatment decision making, and some of the challenges that we're trying to face up to now. And I will just say to finish that this takes a heck of a lot of people, some of whom are named here in terms of the working behind the scenes that makes MCI happen, and really want to finally acknowledge all the patients and families enrolling on this protocol, including our own here at Nationwide Children's Hospital. Thanks for your attention.
All right. Thank you, Elaine. That was an amazing whirlwind tour of all the stuff going on for the MCI. I found it delightful. I liked some of the surprises at the end. Um, I I I would like to encourage everyone to please use the Zoom raise your hand feature if you want to ask questions of Dr. Mardis, and then we'll just take them in the order that hands are raised. And we'll start with Andrea.
Thank you very much. Fantastic talk. I was intrigued also by the zebrafish
Cool,
evaluation.
right?
That's related to my question because I was wondering whether have you also considered perhaps using organoids from these patient tumors to do similar functional analysis? And if not, are there any issues in growing organoids from pediatric patients?
So it's a great question. Actually, I mentioned the German INFORM study that was done a few years ago earlier in my remarks. And there is now ongoing a so-called INFORM 2 study in Germany, and they have adopted the organoid platform to sort of co-screen for the patients as the patient samples go through molecular profiling to really compare the results from molecular profiling as well as the indicated response or not response to, I think, about a 200-ish panel of different therapies, many small molecule inhibitors, some chemotherapies, etc. So I think that we are not doing that as part of sort of this program. I think it will be interesting to learn what they learn. And I think, their numbers are increasing. I advise that program, so I was just there at the end of last year, and the numbers are creeping up. So it's a great idea in principle, I think, to do that clinically as you're anticipating different ways to treat these patients. In terms of challenges, I haven't really heard many. Granted, not every tumor will yield good organoids, and certainly I think the probably biggest challenge is that in some cases for sarcomas, you're really getting just a few needle biopsy passes, so you're going to have to duke it out with path to sort of figure out, whether you have enough left over for organoids. But other than that, I don't think we're seeing sort of like the challenges that we're seeing in PDXs where low-grade tumors just don't tend to develop well in the mouse models. I have not heard of that, but it's not part of our program. There are people that are doing it. Netherlands is the same way. So those data hopefully will be entering into the mainstream, and we may be able to come up with important sort of genomic organoid predictors moving forward for therapeutic answers for some of these kids. Obi, I think, was next.
Hi, Elaine. That was really awesome. I was curious about the one slide you showed that had across your really impressive 4,000 plus patients, there was summaries of the fractions of patients with reportable germline variants and reportable somatic variants, and they're kind of striking on both sides. Like I think, we're learning more and more how much maybe more common these germline variants are than we
Yeah.
maybe were at one point understood. But I was kind of curious if you could explain a little bit more. How does a variant on the germline side qualify as reportable? And then on the somatic side, how do you explain patients that don't have any reportable somatic variants? Like, as I think the numbers were not like kind of 100% or quite close
Got
to.
you. Yeah, so where you're expecting sort of a corresponding percentage. Yeah. So great question. I think the answer to the first part in terms of what's reportable is germline, and Yasmin's on, so she can correct me if I'm wrong. But we essentially coalesce down to about 400 genes that were part of a New England Journal paper a few years back. That was sort of the compendium of currently acknowledged, you know, reportable genes that are involved in germline susceptibility. And then we look carefully at those genes for obvious things like known pathogenic, nonsense, et cetera, um, alterations, as well as for the corresponding second hit, if necessary or if appropriate, in the somatic tissue, is sort of the normal route of doing things. I think one of the things that we've identified is that, and probably this is where the RNA-seq argument starts to make even more sense, is that in some cases there's probably somatic or germline silencing of the other allele that we can't pick up through sequencing DNA, but we actually can see in the RNA that the normal allele, for example, is silenced, and the Lynch syndrome specific known pathogenic variant that's in the germline, now corresponds to a TMB of like 45. So the other thing that I forgot to mention is one of the aspects of the Claret validation that will be new is that we are validating that for reporting out TMB. And so you would be able to have that sort of corresponding piece of information. Why we're missing those, I think, is a question that has probably an easy answer, but it's not easy to get at on a one-by-one basis. And that is, we're just losing some of those variants in the space that is not well characterized by short read technology. And now, we're starting to think about, are there ways to go back and examine through some of these patients in the germline setting, for example, for things that we just can't find from short read or from exome sequencing. I mean, exome sequencing obviously has some blind spots as well. So that would be my answer to why sometimes the somatic and germline numbers just don't match up. That makes sense. Always more to do.
All right, we have 1 minute left, so maybe time for a very brief question from Jay Flynn.
Hello, thanks. Fantastic as always. Yeah, real quick. What caught my eye was the interactive TSNEs in the patient reports.
Mhm.
Now I'm trying to think, what are you projecting and what are you showing? Thank you.
Yeah, so we have we have methylation data classification for a big database of pediatric patients with different diagnoses or different types of CNS malignancies for this particular example. What we project onto that interactive plot, which is zoomable, is essentially those patients are in different colors of dots, and then we have a black dot that corresponds to the patient under study. And you can then localize sort of, if you have more information like variants that may or may not make sense, for example, for that tumor type, you may be able to visualize the methylation status of that patient as being close to, the indicated diagnosis, but maybe on the outskirts, if you will. Or you may be able to see it solidly in the middle of that cluster and basically say, I believe this diagnosis because of X, right? So it's really just sort of more of a research-based tool to sort of look at where that patient fits relative to a bunch of other patients with the same indicated diagnosis. And that can sometimes be helpful.
Very clever. Very clever. Absolutely. It's hard to be absolutely definitive in medicine and all kinds. So close
Yeah.
vicinities matter a lot.
Yeah,
Thank
not
you.
everybody fits into that convenient box, but you have at least some level of confidence that they're relatively close based on the methylation profile. So that's what it's all about.
All right. Well, thank you to again to Elaine for her presentation, for the Q&A at the end, and for all of you for your participation and attention. We'll be back, of course, I think in early March. We're still confirming our speaker, but if not, then we'll see you on March 18th
It's
for a
confirmed.
conversation. It is confirmed. Okay,
Yeah,
great. So we'll be
Kathy and
having the
Karen will
gene,
be giving an update on the gene fusion oncogenicity work group.
as Wes said. And then on March 18th, we'll be having Beth Patel, Dr. Chen, join us for a discussion about the complex variance work group. So we hope to see all of you at our upcoming meetings in March. Thank you again for your attention. See you then.